Good morning, everybody. I'm really happy to be here. Actually, Ela knows this. I've been preaching to the students for a decade that development is where computational neuroscience should head, and I'm thrilled to see that it's really happening. So we're going to start with a session on neural circuit assembly, something very near and dear to my heart. I think many of us go into neuroscience because we want to understand ourselves in some way and understand cognition. It always felt so far away. And I think it's great to start with a talk that shows us that thinking about the human brain, thinking about the primate brain is within grasp. So the first speaker will be Fenna Krienan from Princeton University. She's an assistant professor at the Princeton Neuroscience Institute, and her lab is researching the molecular evolution of specialized brain regions across species. Very cool. She is an NIH Director's New Innovator Award recipient. She is also a Safari Bridge Independence Fellow, a Klingenstein Fellow, and a Pew Scholar. And we're really happy to have you here. So with that, let's go forward.
Thanks, Lisa. Thank you so much, Lisa, and thank you, Ela, for the invitation. Very excited to be here. Sorry, just see. Skip. All right, cool. Okay, so yeah, this is my official title. But I always like to have, an existential title that I put in after. Like, I'm afraid to put it in the program, but the existential title today is not just how do we understand cell types and genetic programs across the primate brain, but now that we put a number to it, what do we do with 5,322 cell types? And I just want to acknowledge that this is not my number. This is the Allen Institute's number, and we'll talk about that. But broadly, my lab is really interested in understanding conservation and divergence of molecular programs across mammalian evolution. And when we do that, we have to contend with, as Ela kind of pointed out, massively different scales, whether it's evolutionary time or the number of cells in the structure or the scale factor in volume and size. So we have these macro level scale factors to contend with, and then we have these types of numbers. And again, this 5,322, where did it come from? It came from the Allen Institute's beautiful study, which gave us the first kind of pass at a mammalian brain. How many molecularly distinct cell types or clusters from RNA expression do we have? And they use a kind of a hierarchical arrangement where they have, the finest level is 5,000, but then they kind of group it up into neighborhoods. And so here's the scale that we're talking about. And all of these cell types are also mapped to space using a reduced sets of genes. So that's the spatial transcriptomics here. So this is a very useful starting point, because now we know the scale of the problem when we start to think about comparative studies of brain composition. But the other thing we know is that, by and large, mammalian brains follow a common bowel plan, if you will. So if you look here at all of the cortical areas, and the only ones that are colored are the sensory ones, and actually motor cortex isn't even on here, all the sensory areas. So what we diverged from is an ancestor that retained these primary areas. They took up most of the cortical mantle. And then through themes and variations, we have relative expansion of maybe higher order areas, but we have the same configuration. So we should expect a lot of constraint and conservation in these kinds of principles. But really what my lab is focused on is when we have the ability to measure individual cell types, and we may be contending with a diversity on the order of hundreds or thousands, really how do different cell types, how do we understand those features that drive the divergence of those cell types? Why do different cell types evolve at different rates? And then focusing on primate lineages, which is kind of our specialty at this point, when and how do they actually arise in development? So that's really what's interesting to us. And when we started this work, we kind of drew up a list. We use a lot of single-cell sequencing, so a lot of the data I'll show you today is based on that modality. And by doing kind of focused comparisons of different brain regions across species, across mammals, what we've drawn up a list of is, the kind of gross ways that we see cell types diverging. So we have changes of abundances, we have changes of genetic programs. The really cool one, I think, is allocating the same cell type, a conserved type, differently across brain structures, because then you can have change to a circuit without changing the base composition, the parts of the circuit. And then, of course, the most provocative and also hardest to ascertain is a true, frank gain and loss of a cell type. So just to give you a sense of what a genetic program change, for instance, might look like, we have, say you only have 4 different major classes of interneurons in the cortex. You can have really qualitative changes where the expression of something like this, which is an autism risk gene, is expressed in one type of interneuron in the primates, and a totally different type of interneuron has this enrichment in the mice. And so that certainly happens, those qualitative changes. But actually what's more common, and I think maybe more interesting to contend with, is the quantitative change, right? So it's not like what exemplifies what we're finding, what exemplifies primate brain cell type evolution is that we have totally different sets of genes that are involved in specifying, the cell types, but rather that we have constraint in the expression levels of the same genes. So if you take the extreme case of loss of function intolerant genes, those are ones that we know are really important because if you have mutations, it's maybe not compatible with survival. Even there, evolution is constraining the levels of those genes in an evolutionarily interpretive way. So I think quantitative expression, rather than qualitative changes, are really what we're kind of grappling with in this framework. The other thing that we've learned, which is humbling, is just the many ways that a single conserved cell type can change. So we have examples where the same cell type, and in this example, it's a interneuron type in the striatum, changes its abundances quite broadly. It also changes its genetic program, so much so that at the beginning we actually missed it in mice. We thought it was a totally different or gain of a cell type in primates. It turns out it's just diverged so much that we couldn't recognize it in the mice. And then the same cell type also, is kind of a wanderer, and it migrates to different brain structures and populates different brain structures across species. So we have cases where even looking at a single cell type, we have evolution playing in all of these different levels. So by way of introduction, what I think I've tried to show is that, we think about cell types diverging in multiple ways across species. What we've become really interested in the last couple of years as I started my lab is how much can we learn about cell type divergence across space in a single species. And so here I'll introduce our charismatic model. This is the marmoset monkey. Of course, Guoping Feng's group is around the corner, and we've collaborated with him for about 10 years. It's been a major source of inspiration for our lab. But we chose marmosets. They're a New World monkey, so they're more divergent to human brains than, say, the macaque monkey, the Old World. It's so a small New World monkey. But the really important feature for us is that it's small. It's about the size of a rat brain. And that means that we can sample many, many, many cells in a given individual. So by the end of the project that I'm going to tell you some stories about, we think we've sampled about 1% of all the neurons in the marmoset brain, which sounds modest, but it's still orders of magnitude more than you could sample with current technologies in something like a human brain, where we have orders of magnitude more cells, and so it's just harder to scale. It's too expensive. And then the other thing that we like about the marmoset is that it fits on a slide. Again, with a rat brain-sized primate, you can actually get the entire spatial context, and that becomes very important for us, especially because I just told you one of the ways that evolution can tinker with cell types is to move them to new locations. So we need to really get the whole picture before we can draw a conclusion about how they diverge. Okay, so we started a few years ago by just taking a census of major brain regions across the marmoset brain. This is before we had even spatial data. And what we noticed is that if you look at the transcriptomic similarities of all of these dissections across major brain structures. Each brain structure has a sort of fingerprint of cell types, so that's not surprising. But when we stared at this, the arrangement of these kind of in terms of their global similarity across all of the brain structures, it seems like there's a fairly simple rule that we can at least move with. And that is that over the course of development, the neural tube closes, it bulges in different places in mammals, and you get expansion of these primary, what are so-called vesicles. And it turns out that where you are along that neural tube will tell you something about which brain structure you're going to reside in. And at the first order, what you can see is that there's a logic whereby cells that came, for instance, from the diencephalon tend to kind of group together, and from the hindbrain to kind of group together. And so that, and this is an adult, right? So there's something retained about the developmental origin, if you will, about a cell type that is retained in the adult transcriptome. So that's useful to us because then we can test these edge cases. Edge cases where, so I should have said, these progenitors who line this developing neural tube, most of the time, as you can see, they will expand into the mature structure, but the progenitors will give rise to the cell types that stay in that structure. So what about cases where they cross over? And those cases end up being very interesting and helpful to us. And so a few years ago, in the mouse, it was shown that there is a progenitor population in the diencephalon. So most of these cell types are going to end up becoming hypothalamic cells. But there's a progenitor population that migrates out of the diencephalon, and it goes into the telencephalon, so the next vesicle over. And they populate a couple of different structures. One of them is the amygdala, the medial amygdala. And this was a very transcriptionally puzzling cell type to us, because when we did the amygdala dissections, they didn't look like other amygdala cell types. And so we're like, why do they do that? Where do they come from? And it turns out that if you just look at their position on this kind of fingerprint map of transcriptional similarity, they actually look very, very similar to their cousins, right? To the hypothalamic cell types that may have shared a common progenitor with them, or at least share a common neighborhood in development. And so what we have come to understand by these kind of rare edge cases is that neurons seem to remember their birthplaces, and those features seem to be encoded and retained in the adult transcriptome. So what is the consequence of that? Well, if that's the global organizational principle of gene expression, then other things can't also be true to the same extent. And one of those things that can be sort of surprising to some neuroscientists, but maybe not at all surprising to developmental biologists, is that when you organize and look at the cell types in this way by their global similarity across all of the brain structures that we had sampled, things like function, if we can just classify that as whether a cell type is GABAergic or glutamatergic, is not the major organizing principle of all of these hundreds or thousands of neuron types, right? You have cell types that express different neurotransmitters but come from the same developmental lineage, and those are more similar to each other. So basically, the lineage is trumping the functional readout if you take neurotransmitter as the readout. So what we've started to appreciate, we and others, of course, is that this global transcriptomic similarity, again, is kind of driven by lineage, and in some sense, it's anatomical context, and not necessarily the functional identity. Okay, so we started in this vein, and then we wanted to expand from a census to a real atlas, because, again, if you have migration and if you have edge cases, you need to know the whole map. And so,
yeah. So what is the reason for the migration? Is that something that you understand? Is it something that, only that hypothalamic area can produce that kind of cell in some way? I mean, you said the functional properties are not so...
Yeah, why does it do that?
Why does it even migrate?
No, I know. We don't know. We don't know. I mean, okay, I should say it is known that the transcription factor OTP is necessary for the migration. But OTP is expressed in all of the other hypothalamic cell types. So why that one, right? Why does that one migrate? And it's conserved, right? We see it in mice and we see it in primates. So we see a few other edge cases of migration. I'll come to one at the end as well in a totally different structure. We love these cases, but of course, we don't see it when they assimilate, in a way. We see the edge cases when they migrate and retain their lineage identity. We don't see it when they migrate and totally assimilate. So it's possible that there are plenty of cases like that too, and we just don't have them. We don't understand what the functional need was for the migration in the first place, but we can read it out. And it's a good question that we grapple with. Okay, so in the study in the data that I'll explain and tell you about now, what we've done is we've kind of taken a more comprehensive look where we take a couple of individuals, we actually register them to an MRI, so we have the whole volume. And then I'm just describing the subcortex, because that's as far as we've gotten. We've kind of left the cortex on the table for now. We take these relatively large dissections, but we make sure that all the edges are next to each other, so we really can say we haven't left any tissue on the table. And then we don't just do RNA sequencing. Now we also add another modality, which is chromatin accessibility. So regions of open chromatin that might be indications of how gene regulatory networks are actually controlling the transcriptomic identity and diversity that we see. And we do the same thing that the Allen does. In fact, this is in collaboration with the Allen, that the Allen did for the mouse atlas, which is that we make a hierarchical kind of tree of cell types. So we have classes, we have groups, and then we have all of these clusters. Separately, we take another animal and we do spatial transcriptomics. With a gene panel of about 300 genes, we map the clusters to the spatial space, and then we kind of explore the spatial context. So this has been a really fun data set to look at. And then I'll just say, again, we have all of these modalities. We directly measure RNA in spatial context in one data set. We directly have joint profiling of chromatin accessibility in RNA in another data set, and then we kind of infer the relationship of space to chromatin accessibility without directly observing it by combining them. So the first question is, well, can we actually do this? Can we take a very large cell type taxonomy, 1,700 clusters is what we have at the moment. How confidently can we assign their spatial provenance in the spatial data? And so the problem with RNA-seq is that we have these coarse dissections. We totally lack the spatial context. We know that these are all in the thalamus, but we don't know where. With spatial transcriptomics, when we map, this is at the group level, so this kind of middle layer of several hundred groups, we actually find really exquisite spatial specificity of these spatially acontextual RNA clusters. And so you can see the lateral geniculate nucleus. You can even start to see the layers. At the cluster level, you see even more layers. The superior colliculus has beautiful layers. The challenge with spatial transcriptomics is that with current technologies, we actually don't have that many genes. So we can define, in principle, this taxonomy using all 25,000 genes, or we can just use highly variable genes, about 4,000, which is actually what we do. But in the spatial case, we only have about 300 genes. So how far do we get with 300 genes? Why do we get such great spatial localization with so few genes? So we were curious about this, and that led us to kind of explore this question in a little bit more detail. So again, we have our taxonomy. And if we take the finest level of this taxonomy, this is all the RNA-seq data, and we map, it's basically just correlation mapping to the spatial transcriptomics in a different donor. And here again, you can see, even at the cluster level, this beautiful laminar organization of the LGN in the primate. And we didn't have the LGN when we developed the spatial transcriptomic custom panel, the gene panel. So we didn't choose the genes based on this data set. We actually chose it based on an older data set that had far fewer anatomical regions. And so we were really curious about why this works as well as it does. So we do a kind of a simple mapping process where we take different sets of genes and we just ask, would we arrive at the same answer in the RNA-seq data if we used fewer genes, and which classes of genes actually matter? So it turns out our spatial panel, which was kind of suboptimally defined for this data set and only contains 300 genes, does pretty well in terms of recapitulating the fine structure that we have in the RNA-seq. Not as well, of course, as taking all 4,000 genes. Not as well as if we had more genes and defined it from the data set directly, but actually pretty well. And so it suggests to me that, again, coming back to this notion of quantitative deployment, there's kind of maybe a reduced code. Maybe you don't need all the genes in order to define all these cell types and in order to map all of the cell types into their spatial context. Maybe small sets of genes, judiciously chosen, are sufficient to retain a lot of to obtain a lot of the performance that we would have had with larger gene sets. And the reason that, and I should say also that this F1 score is pretty conservative, because in fact most of the confusions, so when the cluster gets misassigned to another cluster, it's to its neighbor. So it's to a very similar other cluster. So we actually think we're doing pretty well with a kind of reduced set of molecular features to map these cell types to the brain and to understand their variation. Okay, so we kind of dove into this data set. Of course, we have the entire subcortex. I want to tell you a story about the hippocampus. One of the cool things about the hippocampus in primates is that it has shifted. So in the mice, those of you working in rodents, you might appreciate that the hippocampus has this kind of dorsal ventral orientation. In the primates, in part because of the expansion, particularly of the temporal lobe, we have a kind of rotation of the hippocampus. And so now the axis that matches is kind of the ventral rodent hippocampus to the anterior primate, and dorsal goes to posterior. So with this rotation, the primates have also left something behind. And the structure that is left behind is this tiny structure, kind of vestigial structure called the inducium griseum, which sits just by the corpus callosum, and this is a mouse brain, of course. And it's very, very close to the hippocampus. And in fact, when you do the mapping, and this is the Allen Institute whole mouse brain data, you find that there's a single cluster that populates both the inducium griseum and the CA2 subfield of the hippocampus. And that makes sense because they're developmentally related and they're spatially proximal. And so here's the whole mouse hippocampus. Here are the two cell types. The inducium griseum, again, is outside of the hippocampus, but it sits dorsally near the corpus callosum. Well, what happens when you rotate the hippocampus, as we do in primates? When we rotate the hippocampus, now all of the dorsal stuff is kind of stuck in the posterior side, shifted down. The anterior part is now the ventral part. We've left the inducium griseum behind. It's still up by the corpus callosum. But actually, there's still a shared cell type. We can't partition these two. That exists in the posterior subfield of the hippocampus in the primate, and is also shared, same cells, in the inducium griseum. So when we have this rotation with this vestigial structure left behind around the corpus callosum, we actually retain the transcriptional identity. Well, curious case, we're not sure whether this shift of subfields is a developmental shift or an evolutionary shift. So we're still looking into the evolutionary difference there. But we got kind of excited about the hippocampus because it's this elongated structure and it has a lot of course, functional connectivity relevance along its spatial axis. And so we wanted to look at this in a little bit more detail. What we've learned is that there is, of course, many subfields of the hippocampus, both in rodents and primates. We have the dentate, the CA3s, subiculum and prosubiculum, and there are different cell types, and we can see this at the group level across the anterior to posterior axis. But if we look at a more fine resolution now at our kind of cluster level or finer resolution, we actually see that there is something like a gradient. That cell types, even within one subfield, are assigned to different clusters by virtue of transcriptomic distinctions, and those are arrayed across that anterior to posterior spatial axis, so in the Z plane. So we were kind of interested in what drives and what sets up, what drives, what maintains this spatial organization. If you look just at the spatial data, directly, again, we only have 300 genes, but actually we can find still about 30 or 40 genes that show a very nice anterior to posterior spatial gradient. Some of them are predominantly expressed in the anterior poles and some in the posterior poles. And if you fit trends to this variation to spatially variable genes, we have more or less monotonically and then sloping backwards or forwards in terms of their expression. So if you just look at a single case of FF5, you can see across the anterior-posterior extent of the hippocampus, we have a gradient of this expression. So that was kind of cool. But of course, we only have 300 genes in the spatial data. We're leaving a lot of genes on the table. Like how extended is this code? And so we go then to the RNA-seq data. We infer the median Z position, so the anterior-posterior location, of the major, say, clusters of the CA3 subfield. And we see that they correspond quite well to the spatial transcriptomic order. What we also see is that we can, of course, ascertain far more genes from the RNA data that have this spatial gradient. And what's fascinating to us is that these spatial gradients, you could imagine that when you rotate the hippocampus and you have this spatial gradient, that it doesn't matter which subfield you're in, you share the same spatial gradient. But actually, it seems like the gradients, the genes that are graded, are kind of private, are more likely to be private to a subfield than shared across subfields. So we have kind of selective sets of genes that are arrayed across these anterior-posterior axes within each subfield. So we were wondering, how do you get a spatial gradient? And again, we only have adult, so we're not talking about how do we get it in terms of developmental origin, but how do we maintain it in the adult? And you can imagine a couple of different scenarios. Remember, we have the spatial data, we have the RNA data, we also have chromatin accessibility. You can imagine a scenario where, well, first let me describe the model for anyone who's not so familiar. So typically when we think about target genes being turned on or off, we also have regulatory elements, and these are accessible loci in chromatin that transcription factors can bind to and basically tune the expression levels of these target genes. And so we have what we think of as sort of triplets of transcription factors, the things that bind to the DNA, where they can bind, so whether their motifs are present or absent, and whether the chromatin is actually accessible at those loci. And so if we have these kinds of triplets, can we understand what sets up a gene gradient? So one possibility is that you need a gradient of the transcription factor itself, you need a gradient of accessibility of the regulatory element, and you need a gradient of and then you get the gradient of the gene expression. So that's what we read out, and we're trying to understand which of these combinations sets up the gradient. But it's possible also that just a graded transcription factor would be enough. You could have a permissive regulatory element that it binds to, but because of the concentration of the TF, you get the gradient in the transcription factor. So these combinations are all kind of plausible. If you look classically in developmental biology, you can certainly have cases where just a gradient of a protein or a gene like bicoid can set up a body pattern. You don't need necessarily a gradient in the regulatory element to get the gradient. But there are other cases, like in the spinal cord, where you do need a gradient of the transcription factor. So these possibilities all exist. It turns out in our data, to the best of our understanding, the combination that is most prevalent in these irregulon triplets that we've defined is A, where you have the combination of both. You actually have gradients that we can observe of the transcription factors that are expressed in the subfield. We have gradients of accessibility of the regulatory elements that have the motifs that are associated with the gene expression. And so this is kind of the combination that wins most frequently. This is for CA3, but this is also true of the other subfields, the same general pattern. So just to give one example of such a triplet, we have the gradient of the transcription factor across the anterior-posterior axis. We have a gradient of accessibility of a regulatory element that has those motifs, and then the downstream target gene, in this case FF5, is also graded. Yeah.
I'm wondering for the, you can go back, for the, when you have the graded transcription factor, but not the enhancer, is that more likely for when it's a pioneer transcription factor, actually?
Right, the question is
The top one.
yeah. Especially, the question is would the rule be different if we looked at in development and which one has to be accessible first, or which one is graded first. So you could imagine.
Do you get more pioneers?
We haven't actually looked. I mean, we should look. We haven't looked.
Yeah, okay.
Yeah, but we've actually had that question before. We should just look. We're not sure. Some of the developmental gradients that set up, say, known patterns of connectivity in the hippocampus, like the tenorins, are graded. So we think we probably have remnants of a lot of these developmental pioneer factors, but by no means all. But you're right. We should just look. We should absolutely look.
Can I ask a related question,
Yeah.
much more naive? And
Yeah.
sorry, in the spirit of like bridging between the experimental and the
Yeah.
computational sides, I'll ask computationally naive questions. And so one is, right, is it always a diffusible molecule that is first responsible for setting up a gradient? Because how does, how do you set up a continuous thing without a physical gradient that is diffusible?
I mean, other people should weigh in, but these beautiful classical examples of patterning centers, you start with a gradient. You start with a source, a center, that diffuses along an axis, and that forms the gradient by which that configures either the morphology or the identity or gene expression of downstream genes. So I think the easiest one, the most intuitive one to understand are gradients. Of course, the transcription factors don't leak out of the cell. They are a consequence of some morphogen that is diffusing across space. But yeah, those are the canonical examples that we have.
And just to finish that thought, so you're saying that developmentally, initially, there's a morphogen gradient that may set up, a gradient in like cell identities. But is it the case that to sustain these gradients, you need a sustained morphogen gradient, or then it just becomes the gradients become encoded into each cell's identity?
I suspect, we don't have the developmental data in the primate itself, but I suspect that many of the initial pioneers and the morphogens are no longer present in our data. Yeah.
Yeah,
What do you think?
Add to that I actually think that you can find a lot of cases where you don't actually need like a canonical morphogen gradient, because there's also the timing, right? So we always, in development, the cells are moving. And so it can just be how long they've been exposed. Yeah, so it can be how long it's been exposed, and your prior history can affect how you're gonna respond to something, right? And so morphogens are the easiest to see, but the weird thing is if you actually like look at the proteins themselves, they rarely behave like in a fly embryo where there's no cell boundaries. So it's complicated.
Stas
Especially
has
in an algorithm primate brain.
Yeah.
Another way I like that you can have a graded activity is that you have, for example, this happens in retinoic acid signaling. You have uniform production of something and non-uniform expression of an enzyme that degrades retinoic acid, and in this way, nothing actually has to diffuse, and you establish non-uniform activity of hormonal signal.
It's a really good point, and and it reminds me that I have to tell you that the way that we define these, and by the nature of the chromatin data that we have, I think we are more likely to pick up kind of activator regulatory locations than repressors. It's totally possible that if we had better insight into which features can actually repress gradients, that we'd find many gradients that show the opposite trend of the accessible feature and could act as a repressor. Okay, so I hope we have time today to talk a little bit about gradients versus boundaries. Because one of the things that we know from mammalian brain development is that in some cases you can really think of these cis-regulatory elements, like the chromatin accessibility features that I showed you, which I described as gradients in hippocampus, as quite sharp boundaries in development. And we think of these as really potentially setting up. So after you get your closure of the neural tube and it's all bulged and it's expanded, maybe you no longer have these sharp boundaries, but at least at some point they were present in the regulatory landscape. And so we know that they're active during the progenitor stage. What we wanted to know, because again, we only have adult data at this point, is whether regionally restricted enhancers are retained in the adult. So do you have a kind of adult zip code that can tell you, read out by your chromatin accessibility, that can tell you something about where you came from? And are they shared by separate lineages? And to that, I'll just close on this feature, which also brings in these evolutionary comparisons that we've been very interested in as well. So one of the features I should tell you about primate evolution is that there's a particular structure, the thalamus, that has not itself expanded, but what it has acquired in primates is a population of these inhibitory neurons, a massive expansion of GABAergic inhibitory neurons in the primate thalamus relative to the rodent. So with the atlas, we can also see that there's kind of subtype distinctions across the spatial extent of the thalamus. And the basic question, which has been kind of a question in the field for some time, is where do these guys come from? So it's been known that we have this expansion of this major population, but the developmental origins have been debated. So some possibilities are that because the thalamus is a diencephalic structure, it's part of that vesicular bulge, that these cell types emerge from a shared progenitor with the glutamatergic neurons, which are the majority of the thalamus. But other possibilities are that they migrate in from elsewhere. And so we were interested in looking at this question because of this evolutionary departure. When we do the mapping to the Allen data, just based on the RNA, we see it's quite a striking result that we also see in the human brain, which is that there's a very restricted class of GABAergic neurons in the mouse that map quite confidently to all of the thalamic GABAergic populations in the primate, but also to one that's in the midbrain. And that's no accident. This cell type in the mouse is really restricted to the superior colliculus, which is in the brainstem. So not in the thalamus, not in the diencephalon. So we're interested in this boundary crossing event because it can tell us something about whether you have an imprint both of the gene expression and also of the chromatin accessibility. It turns out that chromatin accessibility, so here we've defined all of our accessible regions across the major brain regions that we've sampled in the data set. And you can see that in general you have chromatin accessibility, just like gene expression, that can be private to a regional zone across these major structures. And when we get to the thalamus, we see something very interesting, which is that specifically in the GABAergic populations of the primate thalamus, you see a shared accessibility fingerprint, if you will, of peaks that are originally defined in the midbrain. So all of this to say is if we collapse this and show it in a different way, the primate thalamus, these GABAergic neurons that have kind of invaded the primate thalamus and are not really present in the rodent may originate from a totally different developmental progenitor population in the midbrain, and specifically in the superior colliculus, but they share their chromatin landscape. They share features of that chromatin landscape with the developmental origin of the population. So we have retention, again, not just in the RNA, but in the chromatin landscape of the cell type. Okay, so I'm out of time. I'm going to wrap up here and just tell you what I told you, which is that anatomic position still remains kind of encoded in a cell's adult molecular identity. When a cell relocates, whether it's in development or evolution, it seems to retain aspects of that birthplace identity, and sometimes that identity can be stronger than anything else that globally configures that identity. What I didn't tell you about, because I didn't have time, but I just want to leave out there, is that you, of course, have cases where cells that have distinct origins can converge. Convergence is a major feature that we struggle with onto a similar molecular identity. So I'll just leave you with one example of that, where we have cells that originate from different progenitor zones. They populate different locations in the adult, but they converge on a shared regulatory program that is so strong that their transcriptomic identity looks like they're cousins. And so we have certainly cases where lineage does not explain the adult form. And those are questions that we're very interested in pursuing. So when and why and how does that shared recruitment of that program happen? So basically, why and when do you trump lineage or trump development in terms of your adult identity? Okay, and with that, I will thank you guys for.
I have microphones here. Do we need them? All right, so then we have 15 minutes for questions was the plan, right? Can I start with one?
Yeah.
So I think it's really interesting, the movement of the cells, and I'm wondering when, you just want to capture.
Yeah, I'm wondering when you look at the molecular differences that define those cell types, how many of those molecules actually could you infer might play a role in just moving the cell, right? Because I'm thinking about, in my mind, it actually makes evolutionary sense. It'd be really easy to repurpose a cell to just move it. And I'm wondering how much of evolution, of complex structures can be explained by just migratory programs.
Yeah, no, I think it's a really potent, it's kind of it's my favorite possibility. And one of the most beautiful examples, I think, in mammalian brain evolution, are not found by us, found by Alex Pollan and Matthew Schmidt, who's now at the Allen, characterizing a type of, so in mice, of course, you have a really expanded olfactory bulb. You have many GABAergic neurons that populate that structure. In primates, you have an expanded prefrontal cortex and many more GABAergic neurons that populate that structure. And so by looking at developmental samples, what they've been able to show is that that cell type that populates the olfactory bulb in the rodent has actually migrated. It's along the migratory route. So you can imagine just a little switch. Instead of going to the olfactory bulb, we barely have any olfactory bulb in a primate, right? Instead of going to the olfactory bulb, just deviate. And it starts to populate the prefrontal cortex. And so they've shown, based on these kinds of analyses, molecular phenotyping, that there's likely a shared homology between those cell types, a really potent redirection that you can imagine have massive functional consequences of expanding the circuit. But what we don't really know is how and when the migration occurs, because we need to do more of these cross-species comparisons to even know that the migration is an adaptation in one species or another. Yeah.
Yeah, I just wanted to say that it doesn't necessarily have to be a migration. So if you look at the nerve cord in vertebrates, we have it on the dorsal side. In invertebrates, it's on the ventral side. And it's a major structure, but it does not involve movement of cells. It involves inversion of the BMP signaling center. So you can have an organizing center that secretes something or expresses something locally that gets things going, which changes how the cascade, how and when and where the cascade is initiated. And this can shift the structure and move cells without actually massive movement of cells. It doesn't mean that migration doesn't happen, but there is a very striking example where it is not involved.
Sorry. Yeah, go ahead.
Sorry.
Super cool talk. So I'm thinking about like the evolution of all these cell types, but also how they can be co-evolving with all the circuitry that they're embedded in. So I'm sort of wondering if you've looked much at wiring genes. I guess you'd have to maybe look a little earlier in development potentially.
Yeah, I mean, that's the major question. The major worry that we have is that all of the wiring specificity is set up in development. We know it's not strictly true. Like the tenurin genes, we can still find it in beautiful hippocampal gradients, and those, of course, we know from Leach and Lowe's work are the kind of relative trade-off of these different isoforms, and paralogs can set up that circuit. So we retain them, but without knowing that in development, we wouldn't have known to prioritize exactly those. But we do think that in principle, moving a cell type can generate brand new circuits that didn't exist. The thalamus might be a really good example of that, and I don't know if anyone in Guoping's lab is here who can speak to this, but they've been looking at the projection mapping of some of the GABAergic neurons in the thalamus, in the primate thalamus, and it seems like there's a projection. I don't know which direction it's in. I guess it's from prefrontal cortex to the thalamus. By definition, that wouldn't be there or wouldn't be prominent at all in the mouse because they lack that downstream partner. So you have a brand new circuit. We don't know, but we don't know, to your question more directly, we don't know what were the molecular features that enabled that circuit to form. Is that a prefrontal projection to glutamatergic neurons that then also just kind of co-opts the existence of nearby inhibitory neurons and also synapses with those as well? We don't know those answers yet, but they're good questions.
Fenna, nice to see you. Very, very nice. So the ability to generate a cell type for a certain part of the brain in another part of the brain gives you a lot of opportunity for variation, for generating a cell that is slightly different, and so on and so forth. So the identity of the cell could be slightly different. Maybe not that different from, it still is an interneuron, it's still GABAergic, whatever. But so my first question is, are they really the same or are they different? The second is that it allows you to control the timing of when cells integrate into the circuitry, the amount of cells that would integrate into the circuitry. Can you comment on those two things? Are they exactly the same? And has the primate brain played with this to alter the proportions and the timing of integration of the cells?
And these are beautiful questions that we struggle with, and again, because we need the developmental data to actually chase some of these chase some of these features. But one thing I'll just go back to with the thalamus question. So in some cases, there are many there's a lot of diversity. It's not just that it's all one GABAergic neuron that migrated into the thalamus and it's all the same. Actually, it has beautiful spatial specificity. Some of the thalamic nuclei have more of this flavor than this flavor. The other thing that we noticed is that there are classes of these cells that really are kind of part and parcel the same as, at least as far as we can tell with our resolution, the same as the ones that are still in the midbrain. But then there's another set that really form its own class. So maybe have further specialized and customized their identity by virtue of coming into the thalamus. We, of course, don't actually know that they arose from a progenitor zone in the superior colliculus in the primate and migrated over. We don't know when that might happen, and we don't actually know if. But we suspect that either there's massive convergence or, more parsimoniously, that they probably arose from a common progenitor. With deeper sequence, with more information, maybe with isoform awareness, we would be able to further. I suspect there has to be customizable details, even within these groups that seem to be kind of spread across structures that we need to learn more about.
Thank you.
Yes.
Yeah, thanks a lot. Very interesting. I appreciate, of course, the approach across different species. I was just wondering, is there enough, potentially enough also genetic variation within marmosets that you can look at different individuals and see that actually they have different patterns of expression, and this is related maybe to difference of their genome?
Yeah.
And then I have a second question, but maybe answer first.
Okay, so the genetic variation is really fascinating in primates because you almost have the perfect kind of middle point between the totally isogenic lab mouse, where you have no very little genetic variation. The populations of marmosets in Guoping's lab and now in our lab are basically as genetically diverse in terms of SNP burden as modern humans. So we have a lot of genetic variation still in the genome itself. But we also have very controlled environments where we raise them kind of all the same, kind of like the lab mouse. So the question that we're very interested in is which one wins? Is it the genetic variability can actually confer variability in gene expression? Or is it that the lack of controlled environments in human populations is really what drives a lot of that variability? And by controlled environment, it could be lived experience, but it could also be the technical features that make human tissue so difficult to work with. We've only done a little bit of this work, but so far it seems like the marmosets are more similar to the mice in that we have kind of very narrow bands of variation with homologous cell types across individuals. But we haven't looked at this enough, and we've only looked at it in neurons. And one of the things I said at the beginning, but didn't go into, is actually neurons are the most conserved case of all cell types in the brain. It's the non-neurons actually that are diverging at more rapid rates across evolution, and I suspect they're also diverging more across individuals.
Yeah, sorry, can I just follow up on this? You were sort of alluding to this already. Do you expect an impact of experience? Like comparing maybe two marmosets, one raised in a very enriched environment and one deprived. Would you expect to see?
Yes,
yeah?
absolutely.
Yeah.
Absolutely. I mean, we're only reading out the end case, the adult case, but I suspect we don't get the diversity if we drastically modify the experience of the progenitors. or the cells as they're wiring. So I think that activity and experience, whether it's cell-cell interaction or actual experience, dark rearing, reduced enrichment, increased enrichment, I think those are necessary conditions to drive the variation that we see. We just don't know how to partition the amount of influence each of those has in the resulting adult taxonomy.
Yeah, thank you so much. I was particularly fascinated by the molecular identity memory of birthplace, and in the end, you kind of compared it to the convergence molecular similarity that you may find. And I was wondering, and maybe there was something in there, and I just didn't catch it. What are the particular components of the molecular identity that indicate the birthplace? So is there something, particular transcription factors or classes of genes or GO terms, that indicate more about the developmental origin and the birthplace and that are different from those that similar between the functionally more compared convergent clusters?
Yeah, these are such important questions. And to tell you the truth, we haven't done enough to figure this out systematically. I mean, one of the things that is helpful is to have more of these, edge cases where we can really identify that the cell type has moved, but it retains some aspect of the birthplace. One thing that's very surprising to us is you think of transcription factors as so important for setting up identity and development. One thing that is surprising to us is that, yes, they are important, but for instance, there are only about 13 of them in our panel of 300 genes in this spatial transcriptomics panel. So we didn't need them to actually map the cell types confidently to in a way, the effect of target genes, the downstream genes of the TFs were sufficient, and the quantitative variation of those were sufficient to pinpoint the locations and the kind of anatomical identity of the cell types that we defined, using all of the genes. So we're kind of grappling with, do we have the right data to really address those questions? The answer is no, not until we have the developmental series. But we're very interested in this question because we actually don't really know the answer yet.
I'm going to take one more question.
Hi, Fenna. Hi. A very striking talk, very, very exciting. So I'm very interested about the developmental origin conservation about these cell types, and the link later on they migrate to different regions of the brain. So as you just mentioned, so I'm about to ask, is there any way we can use the signature you find, these kind of origin, conserve the cell types, and then design some kind of related like enhancer or specific barcoding, then look at their next level, bring connectome change. So we have the origin we have the whole network from the early developmental to later on, how they migrate and connect to the different brain regions from one type, for example, thalamus.
Yeah. No, I think that would be an incredibly exciting direction to move in. And we're thinking about this a little bit. If we could just retain the actual lineage identity of the cell types and then layer on the connectivity. So for example, when the cell type migrates to the thalamus, it may very well be that it's a totally new connectional motif, but somewhere along the way it had to acquire that motif. So did it learn it from its thalamic glutamatergic neighbors, or was it intrinsic to the program, or did it pick it up along the way in development? So we do need those data. What we really love is the connectivity data, the cell type aware connectivity data, because then we can zoom in on these cases where, for whatever reason, the adaptation has kind of proceeded in primates to make a bigger region, more abundant cell type, move it to a different location. We really want to know what the connectivity consequences are of that.
All right, time to move on. Thanks.